What if you could make fabric smoother, brighter, and more sustainable all using fungi? In this video, we are going to explore cbHA from Penicillium chrysogenum, a key gene for cellulose degradation in textiles. Here are our group members. The textile industry increasingly adopts enzymes as eco-friendly alternatives, including cellulase enzymes. Cellulase consists of three main components: endoglucanase, cellulobiohydrolase, and beta-glucosidase. Cellulobiohydrolase plays a key role in biopolishing and stonewashing by breaking down crystalline cellulose from the ends of polymer chains. Its application improves fabric smoothness, reduces fuss, and creates a worn look on denim in a gentler and more environmentally friendly way. This process also reduces the need for harsh chemicals and high energy usage while enhancing fabric quality. The cbHA gene in Penicillium chrysogenum encodes the enzyme cellulobiohydrolase A, or cbHA, which belongs to the glycoside hydrolase family 7. This enzyme degrades crystalline cellulose by cleaving cellulobiose units from the non-reducing end of cellulose chains. cbHA works synergistically with other cellulase to convert cellulose into fermentable sugars, making it valuable in industries such as biofuels, papers, and textiles. Used in textile industry. In textile, cbHA is used for biopolishing, where it smooths cotton fabric by removing surface fibers, reducing fuzziness, and enhancing brightness. It's also applied in stonewashing to give denim a naturally worn appearance. Using cbHA allows manufacturers to reduce harsh chemicals and mechanical treatments, supporting more sustainable and fabric-friendly textile processing.
Next is analysis objective. The objective of gene analysis is to identify the characteristic of the cbHA gene from Penicillium chrysogenum, which encodes the enzyme cellulobiohydrolase A, or cbHA, which is crucial for cellulose degradations. The second objective is to predict regulatory elements such as promoters and the presence or the absence of theta boxes. The third one is to determine the gene's product length and its coding potential, which are useful for optimizing protein expressions in industrial settings. Next is the objective of the protein analysis, which is to predict the secondary and tertiary structures of the cbHA to understand its shape and functional regions. The second objective is to assess the protein solubility using deep soluble E, and this is important for efficient expressions and purifications. The next objective is to analyze potential transmembrane domains and sensory regions. And lastly is to evaluate hydrophobicity, which help to identify membrane-associated regions or surface exposed areas. And then there are three objectives of the phylogenetic analysis. The first one is to determine the evolutionary relationship of cbHA with other species, and then to support the genetic engineering efforts by identifying close relatives as potential sources of improved gene versions. And then last one is to strengthen the foundations for sustainable industrial applications of cbH, especially in textiles, biofuels, and paper industries.
Flow diagram of gene analysis using Softberry. First, open NCBI, then choose nucleotides and search Penicillium chrysogenum cbHA. Click on the first result and copy FASTA. Open softberry.com. On the pop-up menu, choose fgene as H-tool. Paste the nucleotide sequence into the input box, and under Select Organism, choose Penicillium. Click Submit and the result will be shown.
The first step is getting the DNA sequence from NCBI, and then open softberry.com, click search for parameters or functional motifs, and then choose TSSP. And the next step is input DNA sequence from FASTA, and then click process, and the result is obtained. For protein secondary structure prediction using GPRAT4, the first step is getting the protein sequence from NCBI, and then open GPRAT4 server, and the next step is input sequence on the search bar. Next, click make prediction, and the result is obtained.
The tertiary structure of proteins is key to understanding their functions and interactions. The tool used is SwissModel. Start by accessing the site at swissmodel.xpc.org/interactive. Enter the protein sequence and replace the project title with the species name in the format genus_species. Click Build Model to generate the 3D structure using homology modeling. Once the model is complete, take screenshots of the structure. By analyzing the model, we can gain deeper insights into protein behavior across species.
Okay, this is process of identify transmembrane protein from a grape. First, you need to find the DNA or protein sequence from FASTA on RSCB or NCBI, and then input the DNA or protein sequence into the DAS transmembrane prediction server website. Click submit, and the website will start the calculation.
This is the flow diagram to analyze the hydrophobicity of the protein using kite-dot hydropatty plot. The first step is you open NCBI and then choose the protein and then search the Penicillium chrysogenum cbHA and then click the first result and copy the FASTA, and you open the website and choose kite-dot hydropatty plot in the program sections, and then you input protein sequence and then click submit sequence, and you will get a graph as a result.
This is the process of analyzing protein soluble using Deep Solu E. First, open the Deep Solu E website and then input the protein sequence in FASTA format or plain text, and then click the submit button, and the server will start the analyzing. Finally, the result will be shown, indicating whether the protein is soluble or not, along with the probability score.
Okay, next to the flowchart itself. In this process, I first selected the cbhA protein from Penicillium chrysogenum, known as the cellulose. Then I retrieved its amino acid sequence in FASTA format and uploaded it to the ProTart tool at the website itself. The tool generated a visualization highlighting key features such as a signal peptide, an N-glycosylase inside, and several transmembrane helix, indicating a membrane-bound protein. Finally, I saved the image for further analysis and reporting.
This is the flow diagram of isoelectric point and molecular weight analysis. Firstly, we need to open the XPC website and input protein sequence. Click submit, and we will get the output result, including isoelectric point and molecular weight. After that, we need to open the ProtCalc website, input protein sequence, and checklist the isoelectric point. After that, input the charge at pH with isoelectric point results from XPC and click submit. The result will be multiple pH range with charge analysis. To analyze the phylogenetic tree, we need to find other 10 different strains with the similar genes. Firstly, we need to open the BLAST and then choose BLAST nucleotide, paste the FASTA sequence of cbhA, and exclude Penicillium chrysogenum in the species. Next, click BLAST and choose upper 10 result and download the FASTA sequence every file. Copy and paste every sequence to Mega and align with Clustal W. After we finish aligning the other sequence, run the phylogenetic analysis in Mega by choosing maximum likelihood tree. Choose the file alignment, make sure the bootstrap is on, and then run phylogenetic analysis and get the result. We need to repeat with other methods like neighbor joining, UPGMA, and maximum parsimony, and get the other result and analyze the result.
Moving on to gene analysis using softberry.com. From the results shown, it can be known that there are two predicted genes, but no transcription unit and operon. This is because operon is common in prokaryotes, but not usually in eukaryotes like Penicillium. The longest gene product produces is 455 in the first nucleotide sequence.
The result is only one promoter is found at position 372, and there is no data box found. For the LDF, it is very low, it's 0.04, and low LDF will cause low gene expression. To boost cell biohydrolysis production in order to industrial efficiency, modifying with stronger promoter is needed. For the protein secondary structure is predicted using GPRAD4 server. By the result analysis, we can find that this protein is likely a beta sheet-rich structure.
It is the predicted tertiary structure of cbhA, a protein from Penicillium chrysogenum, the fungus famously known for producing penicillin. The model was generated using Swiss model and is shown in a ribbon format, highlighting the overall shape and folding of the protein. This structure represents a monomer consisting of a single polypeptide chain. While many proteins function as monomers, others may form dimers or larger complexes. To assess model reliability, structural validation scores were analyzed. The GMQE score is 0.88, indicating a high-quality prediction. The QMEA and DISCO Google score is 0.85 plus or minus 0.05, reflecting strong structural reliability based on geometric and atomic features. The model was built using a well-characterized template, a cellulose, which shares 73.62% sequence identity with cbhA. This high similarity supports the model's accuracy.
In this analysis, we use the DAS method to predict the transmembrane segment of the cbhA protein from Penicillium chrysogenum strain E2NIAD18. The graph shows several peaks crossing the cutoff threshold, indicating potential transmembrane domains. Among them, the 144 until 156 segments, consisting of 13 amino acids, stands out the strongest candidate due to sufficient weight and score above the cutoff. Another valid segment is 43 until 52, which has a length of 10 amino acid and high GI score of 2.2, suggesting a strong transmembrane signal. However, very short segments like 45 until 49 or 148 until 149 are unlikely to form stable transmembrane helices. Overall, this protein is predicted to contain several short transmembrane domains, suggesting this potential role as integral protein possible involved in transport or membrane interaction.
Okay, so moving to the next one. Here is the protein visualization of the cbhA protein from Penicillium chrysogenum based on its predicted amino acid sequence. As shown in the image, the red colored gradient highlights the transmembrane alpha helix domain, while the green square indicates the predicted sensor domain, specifically an N-glycosylation motif. From the visualization, we can observe that cbhA contains a single transmembrane helix located at the N-terminal region of the protein. This suggests that cbhA is likely anchored to the membrane, with most of its functional domain facing the extracellular space. The presence of the green sensor domain, marked by the N-glycosylation motif, indicates a potential extracellular connection or binding site, possibly for substrates or environmental molecules. This feature supports the idea that cbhA functions in interfacing with the external environment, consistent with its classification as a cellulobiohydrolase involved in cellulose degradation. Unlike receptors like acetylcholine that have multiple transmembrane helix, cbhA only has one, showing a simple membrane interaction. Compared to intracellular proteins like myosin, cbhA has membrane-associated and extracellular features. This matches its role in cellulose breakdown, where membrane anchoring and substrate sensing are important.
Next is how to analyze the hydrophobicity of the protein sequence. Hydrophobicity analysis is a method used in bioinformatics and molecular biology to study how hydrophobic and how hydrophilic each part of the protein sequence is. Analyzing the hydrophobicity of the protein sequence helps predict how the protein folds, which regions are likely buried inside the structure, and which are exposed to water. It is essential for identifying transmembrane domains, signal peptides, and interaction sites. This analysis also aids in understanding protein functions and stability within the cells. In the graph, we can see the x-axis and the y-axis. The x-axis represents the positions of amino acids in the sequence, while the y-axis shows the hydrophily index, which reflects how hydrophobic or above zero and or hydrophilic below zero each region is. In this plot, a window size of 9 is used, meaning each data point represents the average hydrophobicity of 9 consecutive amino acids, offering a relative detailed view of local features. So there are two things that we can see here in the graph. The first one is hydrophobic peaks. Several peaks go above +1, indicating hydrophobic segments. These regions may correspond to transmembrane helices or interior regions of globular protein. For example, a notable peak occurs at the beginning, positions 0 to 2.0, possibly a signal peptide or transmembrane domain. The second one is hydrophilic valleys. The plot dips below -1 in multiple areas, particularly around positions 200 to 250 and 350 to 400. These are likely hydrophilic loops or surface accessible regions. The upper balance, the plot shows a mixed pattern between hydrophobic and hydrophilic regions, suggesting the protein could be a membrane protein with multiple transmembrane domains or a globular protein with alternating surface exposed and buried segments.
So the next one we're going to talk about is the protein solubility. So what exactly do we mean by this? The protein solubility refers to how easily a protein is dissolved in water or aqueous solution without forming clumps or also precipitates. These proteins that are insoluble often form aggregates and also inclusion bodies, which complicate purification and also reduces functional yield. Soluble protein, in contrast, are more likely to fold correctly and also stay dispersed in the solution, and this makes them easier to purify and also functionalize, which is especially critical for the convenient expression. And in this case, we use the tool of degradation because it offers several advantages. The first one is it predicts solubility from an acid sequence using a deep learning approach. And the second one, it is particularly suitable for fungal proteins, like our proteins from the Penicillium. And unlike many other tools, it is not limited to bacterial protein datasets. And it tends to be more accurate for non-bacterial systems such as fungi. And that makes it easier and also great choice for predicting the solubility of the cbhA protein, which is the target of our gene. And here we are using the deep solubility tool that predicts the protein solubility for our target, the cbhA protein from Penicillium chrysogenum. The deep solubility gives a solubility probability score of 0.5684, and since this is above the 0.5 threshold, the prediction indicates that the protein is likely to be expressed in a soluble form, and rather than forming aggregates and also inclusion bodies during the expression. And this is important because soluble proteins are much easier to purify and also use in downstream application, whether for research or also industrial enzyme production. And the question is that why is the cbhA protein predicted to be soluble? The reason cbhA is predicted to be soluble has to do with the properties of its amino acid sequence. First, it is likely has hydrophilic and also water-loving
residues on its surface, and this help the protein interact with water and also stay dissolved. The second, the protein probably falls properly into structures like alpha helicase and also beta sheets, and this kind of folding hides the hydrophobic or water-hating parts inside and keeping the protein stable in the solution. The third, it may contain flexible loops and also coil regions with polar residues. This also help to keep the surface of the protein friendly to water. And finally, the last one, it doesn't show signs of having parts that usually lead to clumping and also aggregation, and that's important because clumping can cause proteins to become insoluble. And overall, these features suggest that CBHAP has a balanced structure that helps it stay soluble, which is what DeepSoluE picked up on.
Next, we will talk about isoelectric point and molecular weight. On this slide, I present the result of the physicochemical analysis of the protein sequence using the efficacy parameter, the isoelectric point or Pi, which refers to the pH at which the protein carries no net electrically charge. For this protein, the Pi is 4.31 and the molecular weight is 48,169.85 gram per mole. From the data, we can see that at pH 4.5, the protein has a net charge of approximately 0, which matches its isoelectric point. At this point, the protein is electrically neutral. Therefore, pH 4.5 is the optimal condition for pH, as protein migrate based on size when their charge is neutralized. Now I will explain the result of the phylogenetic analysis of the gene we identified. This analysis is important to understanding the evolutionary relationship of the genes with other genes of similar function from different organisms. To construct the phylogenetic tree, I selected of total 10 other genes obtained through BLAST search. From the BLAST result, I selected relevant homologous gene to include in the phylogenetic comparison. The first method used is maximum likelihood. This is a statistical method that calculates the most probable phylogenetic tree based on a specific model of evolution. It selects the tree that is most likely to produce the observed sequence data. Its advantage lies in its high accuracy and flexibility, but it requires significant computational power. This method is commonly used when robust, model-based results are desired. Next is the neighbor joining method, which is based on genetic distance between sequences. This method identifies the closest neighbor and cluster them together in the tree. Its main advantage is speed. It is very efficient for large datasets. However, its accuracy may decrease when evolutionary rates vary significant among lineages. The third method is UPGMA, or unweighted pair group method with arithmetic mean. This is also a distance-based method but assumes that all lineages evolve at a constant rate, known as the molecular clock. This makes UPGMA suitable for homogeneous datasets. However, when evolutionary rates differ across organisms, the resulting tree may be misleading. The final method is maximum parsimony, which selects the tree requiring the fewest evolutionary change. This method is character-based rather than distance-based. Its advantage is straightforward biological interpretation. However, it is sensitive to homoplasy or convergent evolution, which may cause similar traits to arise independently. Across all four methods used, the tree structure consistently show the position of cbHA in relation to other functional similar genes. The bootstrap values across all four types of phylogenetic tree generally range from 70 to 100. Typically, values above 70 are considered to indicate strong support, and with many branches reaching value between 93 and 100. The overall tree structure is considered highly reliable. This result also show that cbHA from Penicillium chrysogenum is most closely related to the gene from Penicillium rubens, with a bootstrap value ranging from 99% to 100%.
In summary, the cbhA gene from Penicillium chrysogenum encodes cellulose A, an enzyme vital for cellulose degradation with promising application in textile biopolishing and stone washing, offering a more sustainable alternative to harsh chemicals. Molecular analysis revealed 2 genes, 5 exons, and a weak promoter, suggesting the need for promoter engineering. The protein is beta sheet dominant, soluble, possibly membrane integrated with a high-quality 3D structure, PL 4.31 and MW 48.17 kDa. Phylogenetic analysis confirms its close relation to P. rubens bootstrap 99 until 100%. This research forms a solid basis for optimizing cbHA through genetic engineering for greener industrial use.
This is our presentation. Thank you for watching. Wassalamualaikum warahmatullahi wabarakatuh.